So chapter 12 is about patterns of inheritance. All we're going to be learning about in this chapter is some applications of the concept of sexual reproduction that we learned about in chapter 11. So remember, meiosis makes haploid gametes. When two different gametes fuse together, that makes a new organism. And so we'll be looking at some of the history of how people figured out how inheritance works. And then we'll be looking at some of the common patterns of simple genes. So back in the time of Aristotle, probably even further back than that, people wondered how heredity works. And there were some very common ideas up until the Age of Enlightenment, when people started thinking about things very logically and scientifically. Before that time, people thought that heredity only occurs within a species. So nothing could go beyond that species barrier. And that idea came mainly because the church had, the Catholic Church primarily had dictated that every species on Earth had been created exactly as it is today. And so from that perspective, then there can be no transmission of genetic material beyond that species. The other thing that was thought to be true, which isn't, although it's a technical issue here, is that traits are transmitted directly from parent to offspring. Traits are like your physical appearance, something that you can observe. So if somebody says you have your father's eyes, they don't mean that actually when you were conceived that part of your father's eye somehow transferred into the sperm and then got to you and popped out of your head. What they really mean, which is what we now know, is there's a genetic code that your father has, and when it's translated in his body, it gave him a certain appearance of eye, and you also got that chromosome that has that gene, and you also, it translates to that same shape and color of eye. And so it's not the traits themselves that are transmitted, but rather the genetic code that directs the production of those traits. And so it's not the traits themselves. There was also an idea that traits were carried in some kind of fluid that was sort of drawn into the egg or sperm at the moment of conception, so that there was some kind of blending, like paint blending together between two parents. And we're going to see pretty quickly that that concept doesn't really work when you start testing things scientifically. Another idea, which I really have trouble wrapping my brain around, is this idea that the sperm carried a tiny, fully formed human called the homunculus, and that when the sperm was delivered into the female's womb, it just incubated that homunculus and it just got bigger. I really can't understand that concept, but I didn't live in those days, so in any case, these are all kind of wrong ideas, some of them more wrong than others. What we're going to be looking at in this chapter is the science that led us to our understanding of heredity where we are today. So the first scientist that we're actually going to talk about is Gregor Mendel. And Gregor Mendel worked with pea plants, and I have his book somewhere. Well, this isn't his book, but a book that was written about him. It's called The Monk in the Garden. And but anyway, he worked with pea plants, and a lot of what we attribute to like the basic rules of heredity started with Gregor Mendel. They didn't end with Gregor Mendel, so be careful. He started the field of genetics, and so, but he certainly didn't finish it. So he worked in the mid 1800s. And he worked with pea plants. There were some other, the reason I have this slide up is that there were some other scientists that had already published and figured out the methods for dealing with plant breeding for scientific purposes. And so he was, he benefited from the work that was done before. But let me show you him. Here he is. He was a monk. He was in a monastery that is still there, actually, in a city called Brno, which is in modern day Eastern Europe, Czechia. And he was a monk. And he actually didn't start working with pea plants. He wanted to work with mice, and he was starting to set up mouse experiments. And the story is that a bishop came to visit the monastery and was unhappy that the mice were having so much sex in the monastery that I think it probably smelled bad. He told Mendel to get rid of all of that. So Mendel had to pick something a little more quieter, and he chose pea plants, and he benefited from that other research that showed that pea plants could be hybridized, meaning crossbred in a controlled way. He could get different pea varieties. Peas are pretty small plants, fairly easy to grow. They can be self-fertilized or cross-fertilized. So he managed to collect plants that had ultimately 7 traits. So when I say traits, I'm saying 7. Flower color was 1, seed color was 1, seed texture, pod color, pod shape, flower position, and plant height. That's 7. And then for each of the traits, he had 2 versions, 2 alleles. So he had a version or a variety that only made purple flower plants and a version that only made white flower plants. And those were the 2 versions for the flower color gene, what we would say gene nowadays. Same thing for seed color, 2 versions, same thing for seed texture, and so on. So he collected these plants, and like I said, he controlled the mating. And he learned this from those previous scientists. What you can do with pea plants is, like, let's say this is your purple flower plant and this is your white flower plant. If you wanted to crossbreed them, what you do is you cut off the part of the flower that makes the pollen. So in this case, he cut off from this purple flower plant, he cut off these anthers, which is where the pollen forms. So basically, he castrates the flowers on the purple flower plant. And then he can take the pollen from the white flower plant with a paintbrush, and he can then put it onto the ovary of the purple flower plant. The ovary is this big green compartment. And inside of that ovary, the zygotes will form by fertilization and then grow into embryos. In plants, the embryos are called seeds. So you have to plant the seeds in the ground and wait, and then they can grow up into the next generation. We call those the offspring or the progeny. So the nice thing about Mendel is he had a very certain way of doing things. I think it's probably because he had a very mathematical mind. He was good in physics. In fact, he wanted to be a physics professor, and he went to the University of Vienna twice on a train to interview for a job as a physics professor. The first time he interviewed, they said he had no aptitude for teaching, and so he came back to Brno. And the second time he went to Vienna, when he got there, he chickened out, and so he came back to Brno. And so he was, I guess, he failed at his potential career as a physics professor, but I think we got more benefit that he came back to Brno and then became interested in doing experiments in heredity. So what Mendel did was very had a very certain protocol. He collected the plants I just showed you with all the trait variations. So seven genes, and each gene had 2 alleles, 2 versions. Then he took each plant by itself, and we don't know why he did this. He checked them individually to make sure they were true breeding. True breeding means when you self-fertilize a plant, when you put the pollen onto the ovary of the same flower, it does make seeds, and you can plant the seeds in the ground and grow the offspring. And what he did was he checked to make sure that the purple flower plant only had offspring that were purple flower plants. and the white flower plant only had offspring that were white flower plants. And the green seed plant only had offspring that were green seed plants. And so he checked all of them this way, and anything that was not true breeding, he burned. So like if he self-fertilized a purple flower plant and he grew up the seeds, and some of those new plants had purple flowers, but others had white, he burned all of it. So he only kept the ones that were true breeding. That is key, but we don't know why he thought to do it based on what he would have known at the time. In any case, then the obvious experiment is to cross-fertilize the true breeding strains. The true breeding strains we call the P1 plants. So those are the ones that are true breeding. He tested them, and now he's going to crossbreed them. So he would take the P1 that was true breeding for purple flowers and crossed it to the P1 that had true breeding for white flowers. And then he would get the seeds and grow them, and those would be the hybrids. And when he grew the hybrids, we call those plants the F1 plants. So the P1 plants are the parents of the F1 plants. And then the F1 plants, he self-fertilized those. And the offspring of that self-fertilization is called the F2 plants. So the F2 plants are the children of the F1 plants and the grandchildren of the P1 plants. If plants have grandchildren. I guess they do. So he looked at those F2 plants and counted how many of each variety, how many had purple flowers and how many had white flowers, for example. And he looked for pattern in that data, and he was able to form his laws, which you remember from your preview. He had two laws, the law of segregation and the law of independent assortment. So what I've been describing you is typically called the monohybrid cross. That's one of the first crosses that he did. This is the name of it here. And the monohybrid cross in the P1 generation, or the parental generation, he would take the true breeding purple flower plant, the true breeding white flower plant, cross-fertilize those. And when he planted the seeds, all the offspring are the F1 plants. When he did this, all the offspring came up purple. Even though he knew that one of the parents was white in that cross, all the offspring came up purple. And not light purple, not pink, but like dark purple, the same as this purple parent. So at that point, he could pretty well figure out that blending, let me get my pen here, blending was not going to be the case. There was no blending. It was the full phenotype, the full trait showed up in the F1 plants, the same as it had in one of the parents. But none of the white was coming through on this F1 offspring. So that, I think, probably confused the heck out of him. So he went on and he decided to self-fertilize, like I showed you on the last slide. He self-fertilized. I'll put a little circle back to itself. He self-fertilized those F1 plants, and those plants made seeds, and then he planted those in the ground, and he got the F2 plants. The F2 plants are the offspring of the F1 plants. And when he did that, he counted a whole bunch of them, and no matter how many he had, it always boiled down to a reduced ratio of 3-to-1. 3 that had the purple color flowers for every one plant that had white flower color. And so that also confused him. And so, but he did this over and over and over again with all the different traits. In fact, let me go back to that one slide here. This slide actually shows what the setup was for the P1 parents. And then it kind of skips the F1 generation and shows what he got in the actual data in the F2 generation. So when he did this whole scheme with the purple and white flower plants, he got in the F2 plants 705 purple and 224 white, which he reduced down to a ratio of 3.15 purple for every one white. And when he did the same math with these other ones, he kept boiling it down to a ratio that was really close to what the real ratio is, kind of the ideal ratio, which we call 3:1. 3 that had the appearance of one of the P1 plants and 1 that had the appearance of the other P1 plant. And so that, he knew that meant something because it was happening over and over again. He could replicate it in different pairings with different types of traits. So that's when you can repeat something, when you can show something happens in somewhat different contexts, it means you have something. And also, he never saw any blending with any of the traits. So that leads us to his conclusion. Number one, his plants never showed any intermediate traits, no indication of blending. So, for example, the flowers would always come out either the dark purple or the white, but never pale purple. So no blending. And also, for each pair of traits, he started to notice that one trait would show itself, how do I say this? One trait we call dominant and the other recessive. I'm going to show it to you this way. When he crossed a purple flower plant with a white flower plant, and he looked at the F1 offspring of that cross, all the plants were purple. Even though he knew those plants had something from this white parent, the plant showed only purple, so he called that the dominant. When he crossed a plant that made yellow peas with a plant that made green peas, in the F1, all the plants made yellow peas, none of them made green. So he called yellow the dominant. When you have two alleles that are the same in a plant, there's no real need to use the word dominant or recessive, because if you have two alleles in the plant and they're both for round seeds, then of course the plant's going to have round seeds. If you have two alleles in a plant and they're both for wrinkled seeds, of course the plant's going to have wrinkled seeds. But dominant and recessive, that terminology comes into play when you have a plant that has one round and one wrinkled in each cell, one of each version. And the question is, which one sort of wins? We know it's not blending, so you're not going to get something in the middle. So it's either going to be round or wrinkled, not both. And so the one that's, when there's two alleles that are different in the same plant, the one that shows itself, the one that sort of wins that interaction, we call the dominant. And the one that's sort of hidden in that interaction, we call the recessive. So the gene for the recessive allele is still there, but it's just not showing its observable phenotype at that moment. So he was able to see that there was this concept of dominance. When two alleles of the same gene but different versions, one of them acts in a dominant way, one of them is recessive. So Mendel proposed the following, and I'm using some of his language, but really some of the more modern language. He had to kind of make up words because there wasn't already a vocabulary for this. But the parent plants transmitted discrete factors, we would say genes, that are located on solid particles, we would say chromosomes. The alleles, the two versions of the gene, are discrete. That means they're not liquids, they're solids. So they don't blend together in some way. Each individual receives one copy of a gene or one allele of a gene from each parent. So each individual gets an allele for flower color from one parent and an allele for flower color from the other parent, and they have two alleles then. If the two alleles are the same version, then we already know what the plant's going to look like. But if there's one of each, then one is going to be dominant, one is going to be recessive, and we have to wait and see when the plant grows up what it's going to be. Now, once you do the experiment the first time, like once Mendel did the experiment the first time, it was always reproducible after that. So I don't mean to imply that sometimes white can be dominant over purple. But before he did the experiment, he didn't know inherently which one would be dominant. I know you probably think that darker colors are always dominant, but that's not true. There's plenty of examples where lighter colors can be dominant. And so you kind of have to do, for that particular gene and that particular organism, you have to do that initial experiment and then see what the outcome of that interaction is. And an allele can be present but not express its trait. So that's the concept of the recessive. So Mendel's first law is the law of segregation. I always learned it as law of segregation. Your textbook says principle of segregation, law of segregation. But what this means, segregation is an old-fashioned word for separation. And you'll remember that during meiosis 1, the homologous chromosomes are separated. So you have a gene for flower color on each of the 2 homologous chromosomes. And let's say if you have a purple allele on this homolog and a white allele on this homolog, then in meiosis, those alleles are separated. So that half of the gametes get a copy of the purple allele and half the gametes get a copy of the white allele. If the 2 alleles are the same, if they're both purple, then all the eggs would have a purple allele, or all the sperm would have a purple allele. But depending on what is in the gametes, when 2 gametes randomly fuse during fertilization, that combination of 2 alleles is created in the new organism. If they're 2 of the same alleles, then the offspring will definitely have that particular phenotype. But if there's 1 of each allele, then the dominant will show its phenotype and the recessive one will be hidden. And this is all based on what we know happens during meiosis 1, the segregation of homologous chromosomes. But keep in mind, Mendel was figuring this out with no knowledge of chromosomes, no knowledge of meiosis. None of this had been discovered yet. So for us to look back at it and say, oh, it's kind of simple, it's just sexual reproduction happening in different ways, different alleles coming together, seems easy, but he didn't know any of what we know now. So I'm going to take a break, and on the next section, I'm going to show you a little bit of this logic of how we should think through crosses.